In this video, we will discuss about one of the example of this NK92 based CAR therapy. So here, what they have used is they have modified this NK92 cells with which they call it as this 5.28Z. So basically, this is CAR NK92 cells that can target HER2, which are expressed on the cancer cells. So in this figure, they compared the cytotoxicity effect of this NK92 5.28Z with the wild type NK92 cells against the different kind of breast cancer cells. And the results are shown here for the three different cell lines, that is MDA, MB453, JAMT1 and CAL51. And the difference between these three cell lines is that the first one is trastuzumab sensitive. Trastuzumab is one of the drugs to treat that is used for the patient who are affected with the cancers. So this drugs works in this cell lines, and however in the other two cell lines, that is GMT1 and CAL51, this drugs is ineffective, or basically these drugs are resistant towards this drug. So when they checked this NK92 5.28Z, they are able to see in almost all the three cases enhanced cell lysis or specific lysis of these cancer cells compared to the wild type NK92 cells for all the three conditions. Although the difference in the CAL51 is lower than the difference in the GIMT1, and the difference in it looks like it's similar. It is more in difference is more in the MDA MB453, as the wild type cells does not give any lysis. So this in all together come to the point that by modifying these immune cells, most common cells that are usually modified for this kind of treatment is this T cells, and that therapy is popularly known as CAR T therapies. However, the NK92 cell advantage of these NK92 cells is that it can be used as off-the-shelf therapies. We don't need the immune cells from the patients who are affected with the cancers. We can directly prepare these CAR NK92 cells against different antigens that are commonly expressed in the cancer cells, and can be used at a time whenever the patient is need for this kind of treatment. So this is one of the advantage of this NK92 cells. However, if one wants to use the T cells or the CAR T therapies, then these T cells should be come from the patient to prevent the immunogenic reactions. However, the NK92 cells immunogenic reactions are tolerable. That is shown in the previous clinical trials that are conducted on the patient. So this is one of the advantage of this CAR NK92 cells, that it is not only been used as off-the-shelf therapies, but it can work in cases where commercial drugs are not effective. For example, here the example shown here for the GMT1 and CAL51. So this figure basically shows the opportunities, that is NK opportunities for this CAR NK92 therapies, where people can not only use T cells for modifying it to be more cancer specific, but they can also modify these NK92 cells to be more cancer specific. This will save not only the time that is needed to treat the patient, but it will also reduce the cost of productions compared to the T cells, because in the T cells we have to isolate the T cells from the patient and then modifying it to make the CAR T cells. I hope you understood the results that are presented in this figure, and the different features of this CAR NK92 therapy and CAR T therapies, and the traditional or the commercial drugs that are available. If you need to understand more about this topic in detail, you can go through the reference that has been provided in the description. So that's all for this video. Thank you for your time.